So I'm Harry. I'm PhD candidate at the University of São Paulo in Brazil. We designed a tool to visualize networks over a cell diagram. So basically we have networks, and each node of a network can be annotated with a cellular component or more. In general, these annotations come from gene ontology. So for instance, a protein can be annotated with nucleus, cytosol, and mitochondria. And there are some tools that are simplifying these attributes to better represent the network like in a moment of the system. So two examples are the innate database and the integrated interactome system. The innate database, it simplifies these annotations to show it in the main structure of the cell. That is the nucleus, cytoplasm, plasma membrane, and cell surface, and also extracellular region. It uses the cerebral web to show the network divided into cellular components. Cerebral uses these rectangles, these parallel rectangles, to divide the network. The integrated interactome system, on the other hand, does not have an integrated visualization tool, but it specifies more detailed cellular components, such as the organelles. So when we have a lot of organelles, we have a more complex structure because the parallel rectangles not ever are good positioned. So we ask ourselves if it would be possible to create a more constant cell diagram and draw the network over it, also using a dynamic network layout. So we designed the CellNetVis. This is the main diagram, and they are all the possible organelles we have. So all the organelles that are not in the network, they will be hide. Since the network is based on a dynamic force layout, the user can move the organelles and the network will be repositioned. So the user can look for the better layout for its data. There are some basic interaction functionality, such as changing colors, showing and hiding labels. Also, you can apply edge bundling and change some parameters of force-based layout. If the attribute cellular components is defined, that is, the other possible components a node can be in, it will be highlighted when node is clicked. So the user can see in each other place an interaction can happen. We also implemented the searching and filtering tool. So when the network is large, it's very important, or when the user is looking for a pathway. Since the diagram has his limitations of space, there will be many overlapping of nodes when the network is large. The hairbal problem is also appear here, and so a future work would be simplify the network representation, for instance, using the power graph analysis. On the left, we have an example. The power graph analysis just remove some edges and change the representation of it. For instance, a click that's totally connected can be represented by just a cycle. So this is the main interface of CellNetVis. On the left, you have some sessions for loading the network, changing the nodes colors, changing force parameters, applying edge bundling, and we also implemented a simple diagram as the analysis based on cellular components. It shows the distribution of nodes on the cellular components. On the left, on the right, we have the attributes table. So when you click on a node, the values of its attributes will be shown. On the help page, you can see two demonstration videos, one with a medial network and one with a light network. Also, the data are available there. So, since Heberle is very used by the community, I also integrated the basic layout in the CellNetVis. So, if you want to compare the layouts, you can. So CellNetVis is an independent tool. It's web-based, easy to load, and fast, at least for small networks. You can share the link, for instance, combining the link of the website with the link of the data. It's now integrated in the integrated interactome system. It's freely available and open source. And users that has tested it liked it. And for instance, I have found this tweet, lovely tweet. It was like 2 years ago, actually. It was the first prototype, and I have no idea how that person found my tool. And the person allowed me to show to you this. So I thank all the authors. Also, Hugo Henrique for helping me to integrate CellNetVis in the integrated interactome system, and CAPS and CNPq for funding. Thank you. So if you have questions, you can ask me or send me by email.
Thank you. Also, thank you for catching up with the time schedule. So we have tons of time for questions now, if you have any. Maybe I can kick off.
Hi. I'm personally very interested because I'm developing a very similar tool, and it was nice to see that at the same time something else was coming out in the same direction. I have a ton of questions, actually. So I invite you to come to my poster later if you want.
Okay.
Yeah. But the main thing that I actually didn't catch was, what was the rationale towards the choice of colors for the proteins?
The colors?
Mm-hmm.
I'm sorry, can you repeat?
Yeah, the choice of colors of the proteins inside the visualization. Like you use a certain color scheme, and I was wondering what the colors were based on.
I just have used a color scheme from the color brewer.
Okay.
I just pick one with many colors as possible.
So there's no rationale to, for example, say specific localization has a specific color.
You can change, as the localization is a value of an attribute, you can just change each attribute will be colored.
Okay, I see.
So.
And just the other very quick one is the number of organelles. You actually said that if some organelles are not present in the data that you're loading, they're not going to show up. But what is the upper bound towards number of organelles that you're actually showing? Like you have selected.
I forgot the number, but when a organelle is not in the cell diagram, it will be put on the cytosol.
Okay.
I think it's about 18 or 20.
Okay. So it's pretty much all of them, more or less. Well, most of them. Okay. I really invite you to come later so we can discuss further things that I noted.
Great.
Thanks.
Thank you very much. Very nice role model, I think, visualization for combining functional and spatial organization. I was wondering, currently your tool is connected to a certain database, which is maybe not the highest commonly used database for protein-protein interaction, and I was wondering how generic the tool could be, or at least could be extended to other databases that,
Yes.
you know, have PPI information.
Yes. It's totally client-based, the tool. So it means everything is on your computer. So if you want to integrate this tool, you can on other server. And to load the networks, they are in the XGML format. So for now, if you have a different format, or if the attribute of the localization is not exactly named as it should be, you can open it on Cytoscape and export it. I don't know if it's the answer.
Well, it's a little bit pushing back the ball to the user, which is always something that's a bit of a critical thing, right?
Yeah.
With respect to offering a tool, and then you ask the user to do it himself or
Yes.
herself, right?
Actually, as it's there are I haven't found any other tool that pick one single cellular component for each node. So the user can manually change if
Okay.
in my tool, but it was not good because not always he knows what he is doing. So I don't know.
And the second question, if I may,
Absolutely.
dear chair, is a scalability question. So with respect to what you've shown is, okay, these are, so this example.
Yeah.
So what other ideas do you have rather than just not showing?
Yes. This is very easy to show because the process actually, we don't have overlap here because there is a function to take care of it. But here we need to disable this function, for instance.
Yeah.
So here we have a lot of overlap. And due to the complexity of the processing and due to space. So I think this is the main limitation. But we were surprised that the user can still interact, pushing and moving the organelles. So I think this is a good thing. I don't know if Cerebral could use a force-directed algorithm also, for instance, because here you will have more space with rectangles. Despite you will lose the cell diagram.
Things to work on. Thank you.
Thank you.
Okay, thank you again, Henry. So we have two more